edition of welcome everyone to today's edition of Tissue Talks. The usual reminder to please use the Q&A feature, which is at the bottom right of your screen, to submit questions, which you can do throughout the talk. And then at the end, our speaker will try to address as many questions as possible. So it's my really great pleasure that we have today Adam Engler from University of California, San Diego as our Tissue Talk speaker. Adam trained with another luminary in our field, Dennis Disher, at the University of Pennsylvania, where he received his PhD. And this is the time when he really started to investigate the effects of stiffness of the extracellular matrix on the resident cells and how the stiffness itself regulates the fate and function of different types of cells. He went on with a postdoctoral training with Dr. Jean Schwarzbauer at Princeton University, which was his excursion into the field of molecular biology to build upon on what he has accomplished during his PhD. He was recruited to faculty at the UC San Diego already in 2008. And he was promoted to the ranks, and for the last few years, in addition to being a professor at this department, he's also department chair. In addition, Adam is a resident scientist at the Sanford Consortium for Regenerative Medicine. He's conducting a really interesting research. So some of the key questions that he and his laboratory are studying are how the physical and chemical properties of the niche, of the cellular niche, influence, regulate, or dysregulate the cellular function, and how they modify the genetic mechanisms in both homeostasis, like normal conditions, and in disease. They apply these approaches and methodologies to a number of disease contexts, and in particular, they have a focus on cardiovascular disease, musculoskeletal diseases, and on cancer. Adam's lab is very well known as a place where new materials, new extracellular matrix substitutes are being invented and developed, both of natural and synthetic origin, and with very unique spatial and temporal properties that are designed to mimic the properties of the native extracellular matrix. Their goal is with using these matrices is to improve the behavior of the cells and direct their commitment in vitro and in vivo for either scientific or therapeutic purposes. Adam is very well published and very well cited. He accumulated almost 40,000 citations, and his H-index is an impressive 62. If you look more closely into his published record, you will find that he published some of the really seminal papers. So his first significant paper was the first author paper in the journal Cell in 2006, which accumulated an incredible 16,000 citations, just this one article itself. And this is the article that is reporting how the matrix elasticity can direct the lineage specification of the stem cells. He published in many great journals, including Journal of Cell Science, Nature Materials, etc. And his work continues to really spin around this idea that the physics is as important as is biology for determining the cell fate and function. Not surprisingly, his work has been recognized in multiple ways. He received a number of Young Investigator and Mid-Career Investigator awards from some of the most prominent societies, including the International Society for Matrix Biology, Biomedical Engineering Society, American Society of Matrix Biology, American Society of Mechanical Engineers, and American Society for Engineering Education. Adam is a fellow of the American Institute for Medical and Biological Engineering, AMBI, and he is a recipient of the very prestigious NIH New Innovator Award. Adam, thank you so much for being with us today, and you can now take over the control of the screen.
All right. Well, thank you so much, Gordana, for that very kind introduction. You taught me a few things about myself that I didn't realize in terms of citations and whatnot. But in all seriousness, I really appreciate the chance to come and talk to everyone today about what we've been up to in San Diego for the past several years, and in particular, some work that we've done in the cardiovascular space, say over the last 5 or 6 years. I wish I could bring you better weather today, but unfortunately you can see behind my shoulder here, it's a very overcast day here in San Diego, but that won't rain on the parade for the Padres. I'm sure they're going to do great tonight. So I see some of the members of my lab in attendance, and they know that I'm a big Padres fan. But one of the things that in particular I'd like to share with you today are some of our efforts to understand how the microenvironment can dictate cell responses. And so I'm just sharing in particular this image here created by a postdoc, Tom Molly, in the lab, where we have micropattern cells. The point is to explore asymmetric shapes and how that dictates cell fate in collective cell making, collective cell decisions. And here you're seeing green and yellow are pluripotency markers, whereas red is brachyuria, a mesodermal marker. And so you can sort of see what the distribution of cells are. And the hope is then to make asymmetric patterns of organoids from these shapes, and then investigate how that asymmetry and symmetry breaking leads to differences in the distribution of cells spatially within an organoid. But I won't have time to talk to you about that today. I would just like to start out with a few quick disclosures of companies that have licensed some of the technologies from our lab. And as Gordana mentioned, I just have 2 very brief slides. I have had the pleasure of serving as the chair of the Department of Bioengineering here in UC San Diego for the past 3 years. And one of the first things that we were able to celebrate is the naming of our department. So we received a wonderful endowment from Jean Lay, who is the CEO and former owner of a company here called BioLegend. If you've ever run ELISAs or use a lot of antibodies, you probably used a BioLegend antibody. And so we were very grateful for his support and believing in the vision that I'd outlined. So that has led to a number of different things, including just 2 days ago, we started getting ready for groundbreaking next week for a new $6.5 million instructional lab space for our undergraduate and graduate students. This will be in the basement of our main building here in San Diego, and we're really excited about that. So I just wanted to share that win, along with the fact that our faculty is growing year over year. And so we've been really excited to welcome a number of new and diverse faces to our department, more than doubling the representation of women and as well as underrepresented individuals too. So I'd love to chat offline more with anyone who's interested in spending some time at UCSD or in San Diego. This is just a brief acknowledgement slide so you can see the building that I'm sitting in right now is the Stanford Consortium for Regenerative Medicine. That's the Pacific Ocean right behind us. So we have a lot of first world problems, I like to say, like having to ban things like wetsuits in the lab because people like to surf here. So in all seriousness, though, everyone that's highlighted in bold, in particular, I'd like to thank them just for their contributions to the work that you'll see today, as well as our collaborators and funding sources, in particular, the NHLBI, the National Institute on Aging, and NIBID. We also have a very great partnership here in San Diego with something called the WUSI Human Performance Alliance. They are a great Foundation, the WuSci Foundation is a great organization. They have supported some of the work that you will see today as well. Now, I'm sure that it goes without saying that the extracellular matrix is an incredibly important part of the microenvironment, and that it imparts a variety of different cues for cells to respond to. This classic image from Albert's Molecular Biology of the Cell illustrates how these cells are really interdigitated in a three-dimensional extracellular matrix, and that so the fibers that you're seeing in the image, and that can impart a variety of different intrinsic properties that cells can respond to, whether it's the composition of that matrix, so the matrix ligand that's present, the topography of that matrix, whether you're presenting a smooth surface or a very rough surface, or if the cells are embedded then in that matrix, then it can help dictate cell shape. So whether we're talking about a very tight matrix that's very densely packed and the cells cannot spread very well, or one that has much larger pores where cells are really engaging in 3 dimensions, but also spreading out. All that can really matter, especially to a stem cell. And what we showed now almost 20 years ago and have been exploring that ever since is this idea of matrix elasticity or Young's modulus or stiffness, whatever term you would like to use for this concept. But that's just the difference between, you know, the stiffness that is tissue culture plastic or my desk here versus, say, your body, which is generally not in the in the gigapascal range like my desk is, but is more in the kilopascal range. And that's what's appropriate for biology. And so we want to ask then questions, broadly speaking, about, you know, what does the extracellular matrix have to do with disease, and how is it helping to regulate the cells? So I'd like to start off just with some work that we did now about 20 years ago, looking at animal models of myocardial infarction. And one of the things that we noticed very early on is that the normal myocardium, again, and this is a rat model, tends to be in the tens of kilopascal, so, you know, between 10 and 20 kPa. When an infarct occurs, so when we occlude the left anterior descending artery, all those cells get softer, or I should say the microenvironment gets softer. So as we're measuring it by atomic force microscopy, in the red data there, you can see immediately post-infarct, things get softer. But then over time, we see a massive wave of stiffening that's brought on by excessive deposition, secretion and deposition and assembly of the extracellular matrix by cells in that tissue that have remained alive. And as a result, then it forms a hard fibrotic scar. This process isn't all that different from what you see in things like cancer, or let's say the scar that might develop where you cut yourself really badly and it heals over and is just the tissue properties have permanently changed. So taking this idea and moving forward, we have shown in a number of different applications, as well as many other labs across the world, that cells are going to respond to their microenvironment. And they can also respond when gradients are present. So if you give cells a choice between soft versus stiff, different cells will make different choices. And we've shown, while I think many people have shown that cells will migrate up neurotactic gradients, sometimes you will see examples of cells migrating down those gradients, and that happens in things like cancer. And we've recently published on that, but I won't delve into that data today. What I do want to focus on, though, is a very important and I think really understudied aspect of cell and molecular biology. And that is something we call long non-coding RNA. And these are found in non-coding loci throughout our genome. They are frequent and they are functional. So while we previously, you know, assumed that the vast majority, like 98% of our genomic DNA is non-coding, it's wrong to think that there's no activity associated with it. And about 80% of that has some sort of biochemical activity, whether it's a messenger RNA, a microRNA, something like a small interfering RNA, ribosomal RNA. But I'd also like to posit that these long non-coding RNAs are super important. There are over 28,000 of them that have been sequenced thus far, but none of them, at least circa 2018, none of them were known to be mechanically regulated. So we kind of jumped on this idea and said, well, is there a there? Can we find loci where long non-coding RNA are prevalent, but that there are things like single nucleotide polymorphisms, which are already prevalent in our genome, but are they found in these non-coding loci where they might have some really important function? Now, I will also add the caveat that we are looking for SNPs that are associated with significant risk within these link RNAs. We're also looking for SNPs and link RNAs that are only present in non-human primates or above. So the reason for that segregation, if you will, is that we want to specifically focus in on situations where a mouse model is not going to be effective or it's inappropriate as a system. So we have to use stem cells. So if we look at our genome and we say, where are SNPs that are related to things like cardiovascular disease, and I would draw your attention to the red data here in this Manhattan plot, and you'll see this huge spike around chromosome 9, and those are SNPs that are related to a link RNA called ANRIL. Now, ANRIL is a particularly interesting link RNA. It's contained in this locus 9p21. If you're unfamiliar with this terminology, 9 is the chromosome. P, if we think back to our chromosomes, have a short arm and a long arm. So this is the shorter P arm. And then 2,1 stands for the banding pattern on a karyotype. So this is subband 1 of region 2 within the karyotype of that part of our genome. So this one region in particular happens to be the second largest, what we call gene desert, meaning that nothing coding is in this region. But importantly, the SNPs in this region are going to impart a massive increase of like 2x on early myocardial infarction or coronary artery disease. And it's on par with being a smoker or changes, and it's even greater than changes in age. So we're really intrigued by this. It also is really frequent. So homozygous carriers of these SNPs exist in about 1 in every 4 of us in the human population. So, you know, about 1 in every 4 of us listening today is likely a homozygous carrier of these SNPs. And that can have really detrimental consequences if you are facing, you know, basically a doubling of your risk of heart disease. So we wanted to study this better. And like I said, animal models are not great for it. So one of the first things that we did, and this is a wonderful partnership with Valentina Lasardo, who is now at the University of Wisconsin in Madison, but she used to be here at Scripps in San Diego, along with Kristen Baldwin and Eric Topol. And in getting together, one of the things that we were really excited about was taking peripheral blood monocytes from patient cohorts here at the Scripps Health Network and reprogramming them into induced pluripotent stem cells, but then editing those stem cells to remove the risk region, which is highlighted here in red. Now, why would we want to do this? Why would we want to remove the region? Well, that's because if you look at the outline here in panel A of where the SNPs are, you can see that they're sort of evenly distributed throughout this one region. And importantly, there are so many of them that if we tried a CRISPR-based strategy, there's no way to come up with a clone that would have edited out and corrected all of these SNPs. So we're just going to cut it out and look at risk carriers, so risk wild type in panel C versus the knockout. And then we have a non-risk patient and their knockout as well. And hopefully you can appreciate by this Pearson correlation plot in panel D that there's extreme correlation between each one of these genotypes. So they're clustering based off of whether they're non-risk or risk, rather than anything else. So we were really pleased to see that these cells also are in fact able to reprogram back into stem cells, so they're TRA181 positive, among other markers like SOX and NANOG as well. So as we're trying to differentiate these cells, and we want to differentiate them into all sorts of different cardiovascular lineages, one of the first things that we did was just measure the amount of this link RNA called ANRIL within the 9p21 region, and see how that was affecting the phenotype of these cells. So first, we just need to characterize how much ANRIL is present. And so on the right-hand side, the left-hand side of your images there, you're looking at the long splice variant of this long non-coding RNA. And in the wild type cases, they are expressing full-length versions of the splice variant. In the knockouts, it's not present because that part of the gene, of the link RNA has been removed. Now, if we look at the right-hand panel, this is now measuring the short isoforms of the link RNA ANRIL. And again, hopefully you can appreciate that the knockout versions have very little. Our wild type has a slightly higher amount, but then there's a massive upregulation of this short isoform. And we're going to measure this over and over again and see what is the consequence then of having too much of this short isoform, and whether or not environments are inducing that in the first place. But I will also point out that we don't necessarily see a huge change in the number of genes at the IPS stage. So our bulk RNA-seq is basically showing very few differences that then blows up on us when we start differentiating them. And hopefully you can appreciate in the plot in B that by PCA analysis, we're seeing separation then of our risk cell lines versus all the others, including the edited version of those same lines, so that the isogenic versions are in fact different. So there's a significant amount of transcriptional regulation that is imparted in the cell based off of the presence or absence of a lot of this short isoform, which again relates back to the SNPs and whether or not they're there. So the basic outline then for the next 25 or 30 minutes then, or let's say 25 minutes for questions then, is to take donors. These donors are coming from two different studies that we have here in San Diego. Basically, they're just older patient populations. We take their cells, we do TALEN-mediated genome editing to do the knockout. And now we're at this sort of differentiation stage where we want to put the cells into, or differentiate them either into cardiomyocytes, smooth muscle, or endothelial cells, and then put them into some sort of network, if you will. So the first thing that I'd like to share with you, moving back, is this idea of like a heart attack in a dish. We don't really use that term anymore, but I think that it's cute. But reviewers didn't like that, so we just call it a dynamically changing hydrogel. So we can again take a methacrylated, your favorite gag, but here we're going to use hyaluronic acid. We're going to partially cross-link it at first, so that way the cells are able to bind to it on the surface, and then we're going to fully cross-link it at a different, at a later time point. And we can do this all in multiple sort of sequential steps if we'd like. All the data that I'll share with you today is a single step change. So we're not looking at it in a gradual sense, but we did that in order to see phenotypes really clearly. So if you take home nothing else from today, hopefully it's this slide, which is where we took these risk positive cells. So they do have the SNPs. They have a lot of short versions of ANRIL. And we looked at flux analyses after 5 or so days on these substrates. And basically the cells are fluxing all together. When we do the same experiment, the same cell culture, now all we've done is we've stiffened that environment. So it's gone from 11 to 50 kilopascal. And we're now waiting an extra 5 days. And we come back and we image the same culture. And now all these cells are fluxing at basically the same frequency, but they're all out of sync with each other. So the traces at the bottom here just illustrate the fact that they're that the timing is off in these cells. So why is that? Well, we got really interested in looking at these isogenic lines, and sure enough, they flux in a very regular fashion, even when the microenvironment gets stiffer. So if you look in panel A on the left-hand side, the risk knockout cells, those all flux normally, even when the microenvironment has stiffened, and that's not the case with wild type. Now, we think that this is due in part because the risk wild type cells disassemble their junctions and actually downregulate expression of connexin 43 once the microenvironment becomes stiffer. Now, we won't have time to go into the mechanism, but I do want to share just sort of 2 more slides on this topic. And so the first is, again, confirmation that in our risk patients, we're seeing a higher expression of the short isoforms of this link RNA. If you look at the longer isoforms, there's really no differences between them. So we would attribute these differences to the short version of Anril, especially because in the isogenic comparisons between knockout and wild type, that's exactly what we find. Now, again, this work was published a few years ago, and so I would point you to the reference here, but the really cool part of the story, I think, is the fact that there is switch-like behavior related to whether or not Anril has suppressed this combination of factors P14, 15, 16, which then they themselves have an inhibitory role on the phosphorylation of junk, which then ultimately blocks expression of connexin 43. So as a nod to this, we wanted to see, are there materials that we can use to avoid these sorts of gap junction problems? So we want to fast forward a few years to some work from Giselle Gonzalez in the lab. And what Giselle found was that we can take electrospun polymers. Now, these are P.PSS is what we're doping into the system. The backbone is PVA. And as we selectively increase by weight percent the amount of P dot that's present in the material, again, this is our conductive polymer, we see a massive increase in conductivity. What that ultimately drives for these cells is their differentiation down pathways related to cardiomyocytes. So we see GO terms related to heart contraction, heart process, and muscle contraction that are being upregulated. Now, functionally speaking, the cells that are grown on these 2D substrates then have an increase in their overall calcium flux signal in a dose-dependent manner. So again, moving from left to right in each one of these images, we're increasing the weight percentage of P dots. We decrease things like time to the peak, the full width half max, the cell cycle length. So all these are good signs that are showing or suggesting that the material itself is able to facilitate and propagate these signals from one cell to the next. So what's ongoing in the lab right now, and I don't have time to share with you, is merging these 2 ideas together. So taking our patients that have these connexin 43 defects and adding them into a system like this to see if we can overcome that deficit from a material science perspective. So that hopefully we'll be able to tell you about and share about it in about a year's time. So please stay tuned to that. I'd like to switch gears now and think about our efforts with smooth muscle. And that's a really interesting story because both smooth muscle cells and endothelial cells originate in sort of common pathways and diverge into vascular endothelial cells and smooth muscle. And sure enough, we can make these cells very efficiently independent of their haplotype. So are they risk wild type? Are they risk knockout, non-risk wild type, or non-risk knockout? That does not seem to bother whether these cells are going down a VEC lineage or a VSMC lineage. So A former postdoc in the lab, Jamie Maynor, was able to look at multiple patients and different clones from these patients, and observed that there was, pretty significant heterogeneity even within patients that had the same haplotype. And as it turns out, there's pretty significant variation as well in how they're expressing integrins. But the one thing that I would note is that, in particular, this red population here in panel E hopefully is standing out. That is our risk wild type population. And so despite all the heterogeneity, they actually downregulate integrins related to fibronectin binding, and they upregulate laminin and collagen to a certain extent. So we wondered if we could take some of this heterogeneity that we were seeing in patients, both in terms of morphological differences and also protein expression differences, and see for any one given patient, if they have more cells that are expressing higher amounts of anril or lower amounts of anril, how detrimental is that to them? So one of the other tools that we turned to in the lab was micro contact printing and microfluidics. So we could print our chambers here, and this is just a simple microfluidic channel with the goal of maximizing the inlet and the channel area and maintain specific mechanical properties about the shear stress in these devices. So this is what we came up with, and it allowed us then to expose a significant cell population to shear stress that we could dial in. So I'll draw your attention first to panel A here, and these insets are showing you when we sort these populations in this device, and we set a particular shear stress like 90 dynes per centimeter squared as the selection method, we can create weakly and strongly adherent cell populations, and from that we can then look at their area versus a morphological term like circularity. Now, patient 2 here in red, so the risk wild type patient 2, segregates itself very nicely. We're seeing 96% of the weakly adherent cells that maintain this sort of rounded, small, circular morphology. That was not the case with our risk wild type patient number 1, despite the fact that they had very similar separation mechanics at 90 dynes per centimeter squared. We didn't see the same sort of morphological difference. So we wondered, are these cells just tuned to different mechanical set points? And maybe we could understand some reasons why at a later time. And as it turns out, that was what it was exactly going on. So if you, instead of sorting at 90, if you sort at 30 dynes, now all of a sudden that fraction of the weakly adherent population that is not very well spread, so in the bottom of this plot in pink, that gets bumped back up to 96%, up from 65% earlier. So it seems like these backgrounds can be very heterogeneous, one patient to the next, but even within a single population. So we then again went back to our sorting chamber and we said, okay, can we sort these cells into weakly adherent and strongly adherent fractions and compare that to our unsorted population and see differences? And as it turns out, that was the case. We saw differences in things like the strain energy of these cells from traction force microscopy, the amount of alpha SMA that was being expressed, or integrins. We didn't see changes in doubling time, but what we did notice was that there was a massive upregulation in the short isoforms of anril for our weakly adherent population. Now, to make sure this was causal, we could then adopt a lentiviral expression pattern where our protocol where we take our knockout lines and we reintroduce the link RNA, and we can control exactly how much we're adding by titrating it in panel B, and ultimately then we are able to restore the weakly adherent phenotype by pumping in a lot of anril. So you see this drop from doing nothing in yellow, or in the unsorted population now, the weakly adherent is relatively low. You see that same pattern then with alpha SMA expression as well. Now, that just shows that these cells can be very heterogeneous, but what do they do sort of functionally from the perspective of being in the vasculature? So to answer that question, we then turn to endothelial cells, because we thought that was a much more relevant cell type to work on. And again, we're not seeing differences in their ability to differentiate based off of the haplotype. So baseline, without any exposure to different mechanical environments, the cells are relatively the same, whether they're wild type or knockout. But then we developed a really simple assay, and that was just to create a single channel where we could put in cells that would then self-assemble into what might look like a very premature blood vessel. You can see very nicely the Z01 base junctions there. It is patent because it is hollow, as is shown on the bottom there, and hopefully this will rotate. Yep, so you can see a three-dimensional reconstruction shows that they are in fact hollow, so we can pump fluid through them. So we did just that. We let them sit for about 3 weeks, form a confluent monolayer in these devices, and then we hit them with either an acute bout of low shear stress or an acute bout of high shear stress. And for the acute low, I think everyone can appreciate the fact that there really isn't a change in cell density. There might be some slight orientation changes, but nothing too major going from wild type to knockout. Now, if we were to do the same comparison in the acute shear stress, that's where you really see a lot of changes. So there are many holes that are opening up in these risk wild type cells as a result of being at higher shear stress. So we could quantify that and see that, once the shear stress gets really, really high, then all cells are going to rip off of these neovessels. Whereas if we're sort of in the 60 or so dyne per centimeter squared range, now you're seeing sort of the optimal or the maximal differences between them. So we could take those cells out of the vessels. We could put them then into a sequencing assay and see, are there differences in the transcriptional regulation of these cells? The answer is yes. That's what we're seeing in panel E. And what was really nice was then we could show that very specifically genes that are related to coronary artery disease. So these are studies that are called cardiogram. Cardiogram being an effort to take 10,000 patients with and without CAD, look at commonly expressed genes in their transcriptome, and they identified 88 different loci containing 91 different genes that were differentially regulated routinely between patients that did or did not have CAD. So here we're taking a subset of those genes, and we're showing that in the static environment, the wild type cells are relatively low, and the knockouts are high, but these differences are relatively small, and everything is normalized within each heat map. So when we go to our acutely high shear stress, then you can see the expression pattern flips, and the knockout cells that are making very, very small amounts of each one of these dysregulated CAD genes. Now, the consequence of that, we think, is ultimately, if you go to patients and you look at the walls of their blood vessels, so these are ascending aortas where there was an aneurysm, so we could resect the tissue, stitch the aorta back onto the heart. There's actually several centimeters of give with the aorta, so we can just snip this off and then take this sample for analysis. And whether you are looking in sort of the normal region or the aneurysmal region, there's a huge difference in the thickness of the tissue here. And that's due to the fact that in the risk population, then you're getting a massive upregulation in proliferation as a result then of 9p21. All right. So with that, I would just like to bring us back to this concept that um, the microenvironment really matters in terms of whether you're dealing with a soft environment or a stiff environment, whether you have shear stress present chronically or acutely, and is it high or not. All of these different mechanical influences can be really important to your cells and whether or not they're going to express the things that you want and also show phenotypes that are similar to the diseases that we mentioned. So I would just like to conclude by saying, again, hopefully I've convinced you that these patient-derived iPSCs can be edited to make these very important isogenic comparisons, that materials or the systems that we're using can really reverse engineer processes to understand mechanisms where GWAS certainly just cannot, especially if it's a long non-coding region. And more specifically, that stiffening and shear stress can induce pathological responses in your iPS-derived cardiovascular progeny. So again, this is just sort of an overview of the lab and our workflow within sort of the cardiac space. I just want to thank everyone for their time and attention today, and I'm happy to take any questions as time permits. And thank you as well, Gordana. This was a great, and I appreciate the invitation.
Thank you. Thank you for the really, really great talk. So if you can stop sharing the screen just to quickly introduce the next week speaker. So this will be Ron Weiss from, let's put up the slide. Next one. Okay, Ron Weiss from MIT, who will speak about the programmable organoids, guiding cell fate with synthetic biology and neuromorphic circuits. So we can now start with the questions. I think, Adam, you can also see them in Q&A. The first one comes from Human Falahi. The question is, if you have designed an experiment to determine whether the relationship between cell response and stiffness is causal or correlational. And then there is a little bit more detail that we can address subsequently.
Yeah, no, thank you so much, Human, for the question. So with respect to causal relationships, I would say that, we can put cells on softer or stiffer microenvironments and assess, are different mechanisms changing, and are they changing specifically with respect to just the matrix by itself. I think, what biomaterials have allowed us to do is really decouple many of the other stimuli that I mentioned at the outset today, or intrinsic properties. So we know that it's not the ligand that's present. We know that it's not topography or how porous the environment is, and how a cell's shape is being dictated. So we can take these materials that are very dynamic and then directly probe how they are changing, how the microenvironment is changing as a result of the dynamics, and then see the cell's direct response. We can, essentially knock out a lot of that behavior then, as I was showing here with ANRIL. And so I think that gets sort of at the causal nature and mechanistic understanding of this. Now, I will say one big caveat, I think, to a lot of what people have shown in the literature thus far, is that it's really hard to find specifically what is that protein that is integrating the changes in force and undergoing some sort of deformation that introduces some sort of new signaling cascade. I think a lot of us are close in a variety of different contexts, but it is very difficult to sort of pinpoint one particular protein and say, this is the protein that is responsible for converting physics into phosphorylations. We had shown this about a decade ago in muscle with a protein called vinculin, where there's a cryptic phosphorylation site that opens up under force, and the cells tend to apply just the right amount of force to vinculin as that protein then opens up and exposes this cryptic site. So that's sort of my hand waving response to say that I think there is some causal data out there. It is just hard to specifically pinpoint the physical into phosphorylation event.
Okay.
Sorry, I'm trying to read the rest of the question, but
Yeah, so I was wondering if you already, I think you addressed this at least in part, but maybe you can elaborate a little bit more about the methodological tools that we have today to really parse out the effects of chemistry from the effects of stiffness, because the question is also like, what if you achieve the exact same stiffness of the matrix using different chemistries and vice versa? You know, how much would it affect the phenomena that you're studying in terms of like
yeah.
responses of cells to the physical factors?
Thank you very much, Gordana. It's a wonderful summary of my rambling explanation. There was that one of the things that we did as well about 7 or 8 years ago was to look at systems where you basically have a polymer, a long chain polymer and a crosslinker, and you want to put them together and form a network. They're essentially two different dials that control the same thing, which is the mechanical properties of the material. So what is its Young's modulus? So you can achieve them in two different ways. And one of the things that we showed in a Nature Materials paper a number of years ago now was that you can use more crosslinker or less and compensate with more of the polymer and achieve the same mechanical properties, but then also achieve the same outcomes with those cells. So they differentiate to the same extent, which I think showed that mechanics then is really important, and it's sort of less so the specific polymer to crosslinker ratios that people are using that might dictate things at a very nanoscopic level, like how porous is the material, but they're not porous at the length scale that, at least these hydrogels are not porous at the length scale for a cell. So I would argue that while chemistry is really important in terms of what protein is on the surface, how is it being displayed, is it tethered or just physisorbed onto the surface? What seems to be the determining factor outside of growth factors and all of that is the mechanical properties of the environment.
So maybe this is like a good point, maybe for just one more extension. You talked a lot about stiffness. Question is about stiffness, but now, as you say, mechanical properties, like how much more complexity we need to add to the analysis of biomechanics, because it's obviously more than stiffness. I mean, it's viscoelasticity, it's, like many. So what is your
Yeah.
about what really matters? What is it that the cells are sensing in their environment?
I'm so glad that you said viscoelasticity, because I don't want to sound like a one track record or a one hit wonder. We are certainly cognizant of the fact that cells don't just pull and sense elastic properties, but they can grab onto and hold materials. And when materials are viscous, that there can be some dissipation there or stress relaxation, and that can lead to different outcomes. Now, I will certainly admit that just looking at the influence of elastic properties is less complicated, and a lot of engineers, but also of even bigger community of cell biologists have really sort of taken this on and run with it. And there are a number of companies that sell these gels now, and I think that it's great, but the story is definitely much more complicated. And so we've been very recently looking at gels similar to what like Ovi Chowdhury and Dave Meany have shown, where the viscous dissipation is really important for the outcome of those cells. And so I think we're beginning to see in our own work that there are important interactions with more viscous components of the microenvironment that we haven't previously talked about. So I think that work is certainly ongoing. And I think that it's really extremely important, as their work has shown, in situations like breast cancer. You can get differences in how cells migrate and their integrins interact and engage with the microenvironment. So again, I don't want to ramble too much, but I think that's something that's really important. And we will, as a field, I think, be tackling those ideas very soon. And hopefully, just as elastic properties kind of got bought up by the cell biologists in the room, hopefully we can see the same thing with viscous properties too. Ooh, I'm sorry, I think you're muted, Gordana.
Oh, I'm sorry. You have a question from Dilip Thomas. So he is interested if you see differences in connexin 43 expression between knockout and control lines, but after differentiation, after the cells.
Correct. Yeah. So the connexin 43 difference is limited to the risk wild type cells. So the knockout cells maintain their connexin 43 expression even when the environment became stiffer. And the wild type cells lost their expression because they have upregulated ANRIL.
Yeah. And then like the sort of a follow-up is, do you think that this can explain the coupling issues that you observe following stiffening?
Yeah, no, I'm happy to follow up, Dilip, if you want to send me an email. We had several other control experiments in that paper that I think very nicely answer your question, where you could see disruption in gap junctions, so we could sort of mechanically perturb them, cut the cells in half, and you'd see like the signals would propagate and then stop where we disrupted a monolayer, let's say. So lots of work that I didn't get a chance to share, and I'm happy to talk about that offline.
Okay, very big question from Sheravanti Homer Vaniaskian. I'm sorry if I didn't say it right. Can you recapitulate the atherosclerosis in a dish?
Ooh, I know that many people are trying. We have not specifically tried to do that other than by changing the flow rate into our systems and thus increasing the shear stress on the vessel wall where our cells are. So I think many people are much further along than us, and we are really focused just on developing a simple assay where we can see differences in function with our 9p21 cells. So I think the really short, naive answer is yes.
Okay.
But I point to others. Yeah.
Okay. Question from Onur Aydin. So he is referring to the communication between cardiomyocytes, which is a known phenomenon. This is how they synchronize their beating. So it's mechanical communication leading to this. And then substrate is obviously playing a big role. So can you comment on like what do you see as the major role of this mechanical communication, cell-cell communication in the context of mechanical factors in the environment?
Yeah, and thank you so much for the question. So certainly differences in the propagation of strain will be present in the 11 versus the 50 kilopascal. So you would expect the strain field to be vastly different. That's not to say that the cells could not or did not deform the microenvironment. It was just more muted. So I don't know to what extent, and we have not specifically looked at what is the distance over which these cells can communicate, and I mean the cardiomyocytes. We have asked that question with skeletal muscle, where we looked at how aligned the skeletal myotubes were, the closer together that they were, or the further apart that they were. So it seemed like about 3 or 4 cell widths was about the distance over which these strains would propagate. That was, of course, in an environment that's about 11 or so kilopascal for normal muscle. What that looks like at 50, I'm not quite certain. But I wouldn't say that it's no longer a dominant mechanism, because these cells were still able to cycle their calcium correctly. It was more that they were just out of sync with the cardiomyocyte example. So I think I would push back just a little bit and say I still believe that it's important. It's just that the synchronicity was our point there.
Okay, and maybe like we are nearing the top of the hour, maybe you can take one more question, and then we will ask you, we will give you contact information if you could answer some of those that we didn't have time for, just directly to the attendees.
Sure.
So the last one is by Christopher Solis. The question is it that there are too many proteins that are being activated simultaneously, and then they sort of talk to each other? He even mentions integrity effects. So it is it's really hard to pinpoint, like what is the main cause of a specific event. If you can just comment on, like, I'm not even sure how to formulate the question, but like how to approach the fact that it's not as simple as it may.
So in full disclosure, Chris and I have known each other for a while. He's actually part of a program that we call Focus here at UC San Diego. It's a great junior to senior faculty mentorship program. And Chris is my mentee. So it's good to at least digitally see you today, Chris. And thank you for the question. I did not prompt him, so I just want to put that out there. Indeed, it is a really hard problem to think about, and I'll offer sort of 2 unanswered questions that we're still trying to deal with. The first is, as you mentioned, there are likely multiple signaling pathways that are redundant, such that if you were to knock out any one signaling mechanism, others can compensate and achieve very similar things. We actually see this in breast cancer. So we did a study a few years ago, again with these dynamic materials, where when we just had static environments, you saw one set of signals come up. When the environments were dynamic, you saw a slightly different set of signals that were becoming activated. And if you interrupted in the soft environment any one of these signals, you could prevent mechanosensing. If you did that in the dynamic environment, the other pathways just compensated for the signals that were lost. So it's a very difficult scenario where it might not just be one protein that's converting the biophysical into a phosphorylation difference, but multiple pathways that can compensate for each other. And so that makes this a very difficult problem to tackle. So I think that it's a great question, one that we are struggling with. And I would use the breast cancer example from our, we had a PNAS paper like 2019 or so, where we saw this effect, and it was really kind of infuriating.
Thank you, Adam. I think this is like a good comment, closing comment. Thank you again. This was really awesome. And like any great research, you always, like anything you find out, opens 70 new questions, and your talk and this discussion are really great examples of this. So thank you. And thanks everyone for very nice, insightful questions. So hope to see you next week.
Thank you so much.